Okay, we have been done in an experiment to determine the concentration of the NaCl in isotonic, hypertonic, and hypertonic, and also establish the effect of the different concentration of NaCl solution in the electrolyte, and also determine the lysis point of the electrolyte, and also determine the penetrating and non-penetrating solute. This is the equipment and the material that we have been using in this experiment. So you can see this is a list down that we have been used.
Now we move on with procedure. Section E1. Prepare for test tube label hypertonic, isotonic, hypotonic, and distilled water. Add in 5 ml of corresponding solution into each test tube. Next, add 15 microliters of blood into each test tube by micropipette. Then, cover each test tube with parafilm, tipping all the tube gently and incubate at room temperature for 20 minutes. Mix them well every 5 minutes. Then, centrifuge all the tubes for 5 minutes at 2000 rpm. Make sure all the tubes is in balance. By using transfer pipette, transfer the supernatant into clean test tube and measure the absorbency of supernatant. Section E2. Prepare 12 test tubes and label accordingly as 1, 2, 3, 4 until 12. Add in 5 ml of 1.0% sodium chloride solution into test tube 1 and add in 5 ml of 0.85% sodium chloride solution into test tube 2 and so on until test tube 12. Add in 5 ml of deionized distilled water. Repeat procedure as in section A1 accordingly. Then plot graph. Section B. Prepare 8 test tube, label 1, 2, 3 until 8. Add 5 ml of 300 mosmol sucrose into test tube 1. Add in 5 ml of 300 mosmol glucose into test tube 2 and so on until test tube 8. Add in 5 ml of deionized distilled water. Repeat procedures as in section A1 except incubate the test tube for 20 minutes instead of 20 minutes.
Okay, this is the results for our experiment. So for section 1A, which is to establish a reference for the next experiments, we have the control is distilled water and we give it a percentage hemolysis of 100%. So the absorbance is measured using a spectrophotometer, and this is the formula we use to get the percentage hemolysis, which is absorbency divided by absorbency for the control times 100. Okay, for section A2, we have different concentration of sodium chloride, and again distilled water as the control, and this is what we get. Okay, for section B, we have different solutions such as sucrose, glucose, sodium chloride, urea, and again distilled water as the control with 100% hemolysis. And finally, we have the graph that is plotted for percentage hemolysis against sodium chloride concentration. This is from section A2. So you can see that the percentage hemolysis will decrease when the concentration increase.
Okay, here is a table that we can see is a hypotonic, and also the hypotonic means that the water concentration in the solution is higher and the water concentration in the cell is lower, while the water from the extracellular fluid will move into the cell and then causes the cell become swell and also burst. The hemoglobin in the red blood cell or the erythrocyte will move outside to the solution to form red color. And also the isotonic, which means that the water concentration in the solution is equals to the cell, and the cell should not undergo any change. While the hypertonic means that the water concentration in the solution is lower, while the water concentration in the cell is higher. The cell will become shrink and undergo crenation because of the water in the cell is moving out to the extracellular fluid due to the water in the solution is lower, and also causes the hemoglobin does not leak out or release out to the extracellular fluid, or causes the solution become clear. Thus, the hypertonic percentage of hemolysis, this one is larger compared to this one and this one. Well, this one compared with this one is larger because the percentage of hemolysis at here is larger compared to this one. So this one is less percentage of the hemolysis because the cell is shrink.
In experiment A2, the color in hypotonic solution, which is sodium chloride with concentration from 0.5% into to 1.0%, is clear. This is due to the low permeability of membranes towards hemoglobin. The hemoglobin cannot move out from the cell because of the force do not enough. Therefore, hemolysis did not occur. And from the graph plot from section A2, we can conclude that in general, the percentage of hemolysis is decreased when concentration of sodium chloride is decreased. And in this experiment, the concentration of sodium chloride solution from 0.45% to 0.1% turned to red color due to the high permeability of membranes toward hemoglobin, and the cell undergo hemolysis. Thus, we can say that the concentration that lowers than 0.5% is hypotonic solution, and the concentration that higher than 0.5% is hypertonic solution, while the concentration for 0.5% is isotonic solution.
In section B, we have the first test tube. The extracellular fluid is a 300 mmol of the sucrose, where the intracellular fluid is a 300 mmol of the non-penetrating solute, where the sucrose is a non-penetrating solute. So the total of the osmolality for the extracellular fluid is 300, where then in the intracellular fluid is a 300 mmol. Where then both of the same causes is isotopic, where then the water is equilibrium due to the non-penetrating of the extracellular fluid, and the intracellular fluid is the same. So it's isotonic, and then the no change in the volume of the cell thus causes the solution in the test tube 1 become colorless. In the test tube 2, the extracellular fluid contains of 300 mmol of the glucose, and the intracellular fluid contains of 300 mmol of the non-penetrating solute, where then the glucose is the penetrating solute, and the total of the osmolality is 300 mmol in the extracellular fluid, which is equal to the intracellular fluid, also have the 300 mmol of the total osmolality, causes it to become an isomosmotic. And then the extracellular fluid don't have the non-penetrating solute, but the intracellular fluid have 300 mmol of the non-penetrating solute. Thus, the tonicity of the intracellular fluid is higher than the extracellular fluid, causes is hypertonic, where the cell will become swelled due to the water is moving into the intracellular fluid, and the hemolysis will be occur, causes the solution in the test tube 2 will become red color. In the test tube 3, we can see the extracellular fluid we have 300 mmol of the NaCl, and the intracellular fluid have 300 mmol of the non-penetrating solute, where the NaCl is a non-penetrating solute, and the total of the osmolality is 300 mmol in the extracellular fluid, is equal to the intracellular fluid of the total osmolality, because this is isoosmotic, where then the non-penetrating in the extracellular fluid is equal to the amount of the intracellular fluid, which is 300 mmol, causes is isotonic, where the water will be moving in and out, and causes no change in the cell volume, and the solution will become a colorless. In the test tube 4, the extracellular fluid, we have 300 mL more of the urea. Intracellular fluid, we have 300 mL more of the non-penetrating solute. But then urea, we know that it's a penetrating solute, and the total of the osmolality in the extracellular fluid is 300 mL more is equal to the intracellular fluid, also have the 300 mL more, so it's called isoosmotic. But the extracellular fluid don't have the non-penetrating solutes, where the intracellular fluid have the 300 mL more of the non-penetrating solute. So the tonicity of the intracellular fluid is higher than the extracellular fluid because it is a hypotonic, and the cell will become swelled due to the water from the extracellular fluid is moving into the intracellular fluid, and the hemolysis will occur, and the solution in the test tube 4 will become red color. In test tube 5, the extracellular fluid will have 150 mL more of the urea, and the intracellular fluid will have 300 mL more of the non-penetrating solutes, where the urea is the penetrating solute. Okay, the total of the osmolality is 150 mL more in the extracellular fluid, where then total osmolality in the intracellular fluid is we have 300 mL more, which is higher than the extracellular fluid, because it's considered a hyperosmotic, where then the extracellular fluid don't have the non-penetrating solute, where the intracellular fluid we have 300 mL more of the non-penetrating solute. Thus, the tonicity of the intracellular fluid is higher than the extracellular fluid, because it is becoming a hypotonic, while the cell becomes swelled due to the water from the extracellular fluid will be moving into the intracellular fluid, and hemolysis will occur, and the solution in the test tube 5 will become red color. Test tube 6, we have extracellular fluid with 600 of mL more of a urea. Intracellular fluid, we have 300 of mL more of a non-penetrating solute. But then the urea, we know that it's a penetrating solute, and the total of the osmolality in the extracellular fluid is 600 mL more, which is higher than intracellular fluid, which is 300 mL more, which is a hyperosmotic. But then the extracellular fluid don't have the non-penetrating solute, where the intracellular fluid we have 300 mL more of the non-penetrating solute, where the intracellular of the tonicity is higher than extracellular fluid, and then causes is a hypotonic, and the cell will become swelled due to the water of the extracellular fluid moving into the intracellular fluid, and the hemolysis will occur, and the solution in this test tube 6 will become red color. In the test tube 7, extracellular fluid, we have 300 mL more of the NaCl and 300 mL more of the urea. And the intracellular fluid, we have 300 mL more of the non-penetrating solute, where we know that the NaCl is non-penetrating solute and the urea is the penetrating solute, where the total of the osmolality in the extracellular fluid is 600 mL more, which is higher than intracellular fluid, which has 300 mL more, and this is called the hyperosmotic. But then the extracellular fluid, we have 300 mL more of the non-penetrating solution, solute, which is NaCl, and equals to the intracellular fluid of 300 mL more of non-penetrating solute. So the tonicity of the intracellular fluid and the extracellular fluid is the same, causes is isotonic, so no change in the cell volume, so the water is moving in and out, and the solution in the test tube 7 is become colorless.
Precautions that should be taken when conducting the experiment are: hand gloves must be worn when dealing with blood to prevent blood contamination. After covering each tube with paraffin, the contents must be mixed well to obtain homogeneous solution by dipping the tubes gently and carefully. All the centrifuge tubes should be labeled accordingly to prevent confusion. The time needed to leave the tubes at room temperature must be followed accurately to prevent inaccuracy of results. The container containing supernatants must be cleaned with tissue paper first before putting it into the spectrometer. What we can conclude from the experiment is the movement of water molecules is affected by the tonicity of solution and does not depend on the osmolarity of the solution. The erythrocytes swell in hypotonic solution, crenate in hypertonic solution, and no changes of volume occurs in isotonic solution. The percentage of hemolysis is inversely proportional to the concentration of sodium chloride solution. The water movement only refer to the extracellular non-penetrating solute concentration to the cell's non-penetrating solutes, because penetrating solutes will cross the membrane and reach chemical equilibrium. The isotonic solution for erythrocytes is 0.9% of sodium chloride solution.
In this experiment, we have learned that the tonicity and the osmolality, where the hypertonic, isotonic, and the hypotonic is determined by the color of the solution in the test tube, where the penetrating is the glucose, urea, and urea, and the non-penetrating is the NaCl and the sucrose. In this experiment, we have been known the penetrating and non-penetrating solute. Well, in this experiment, we have been known that we have to alert and caution to certain things, such as we need to wear a glove when we transfer the blood with a micropipette. And more detail of the osmosis is by conducting a practical and experiencing it with our own self.